Welcome, everyone. Today, we'll explore the fascinating and increasingly urgent topic of antibiotic resistance mechanisms. By the end of this lecture, you should be able to do several things. First, define antibiotic resistance in clear terms. It's more than just treatment failure. It's a dynamic evolutionary process. Second, identify and explain the major mechanisms by which bacteria resist antibiotics. We'll break these down into enzymatic inactivation, target site alteration, efflux pumps, and changes in permeability. Third, we'll look at real-world examples. You'll see how different bacterial species employ these mechanisms in both clinical and environmental settings. We'll also examine how resistance genes spread through plasmids, transposons, and horizontal gene transfer. This genetic mobility is key to the rapid global rise of resistant infections. Finally, you'll be able to critically evaluate strategies for tackling antibiotic resistance, including stewardship programs, novel drug development, and even emerging technologies like CRISPR. Think of today as both a microbiological deep dive and a strategic briefing. Resistance is one of the top public health threats of our time, and understanding the biology behind it is essential for anyone working in medicine, microbiology, or public health. Let's begin. By the end of this lecture, you should be able to do several things. First, define antibiotic resistance in clear terms. It's more than just treatment failure. It's a dynamic evolutionary process. Second, identify and explain the major mechanisms by which bacteria resist antibiotics. We'll break these down into enzymatic inactivation, target site alteration, efflux pumps, and changes in permeability. Third, we'll look at real-world examples. You'll see how different bacterial species employ these mechanisms in both clinical and environmental settings. We'll also examine how resistance genes spread through plasmids, transposons, and horizontal gene transfer. This genetic mobility is key to the rapid global rise of resistant infections. Finally, you'll be able to critically evaluate strategies for tackling antibiotic resistance, including stewardship programs, novel drug development, and even emerging technologies like CRISPR. Think of today as both a microbiological deep dive and a strategic briefing. Resistance is one of the top public health threats of our time, and understanding the biology behind it is essential for anyone working in medicine, microbiology, or public health. Let's begin. Antibiotic resistance occurs when bacteria evolve the ability to survive exposure to antibiotics that would normally kill them or inhibit their growth. It's important to understand, bacteria, not humans or animals, become resistant. This is a natural phenomenon. Bacteria have always had resistance genes. We're just accelerating the process through overuse and misuse of antibiotics in medicine, agriculture, and even everyday products like soaps. Resistance can be intrinsic or acquired. Intrinsic resistance is built in. For instance, gram-negative bacteria have an outer membrane that makes them naturally less permeable to certain antibiotics. Acquired resistance, on the other hand, happens through mutations or by picking up resistance genes from other microbes. Why does this matter? Because it turns once treatable infections into serious, even deadly, threats. Diseases like pneumonia, tuberculosis, gonorrhea, and foodborne illnesses are becoming harder to treat. The World Health Organization has declared antibiotic resistance one of the biggest threats to global health. And it's not just about healthcare. It affects food security, economic development, and global trade. So when we say antibiotic resistance, we're talking about a biological adaptation with massive public health implications. Let's take a bird's-eye view of how bacteria resist antibiotics. There are 4 major mechanisms. Enzymatic inactivation. Bacteria produce enzymes that chemically destroy or modify the antibiotic. Think of it like cutting the red wire on a bomb. It deactivates the threat. Target site modification. The antibiotic's target, like a ribosome or enzyme, gets altered, so the drug can no longer bind effectively. It's like changing the lock so the key no longer fits. Efflux pumps. These are proteins that pump antibiotics out of the bacterial cell before they can reach their target. It's like ejecting a poison before it has a chance to work. Reduced permeability. Some bacteria reduce the number or size of porins, making it harder for antibiotics to get inside the cell. These mechanisms aren't mutually exclusive. Bacteria often use multiple strategies simultaneously, especially in hospital settings where multidrug-resistant organisms thrive. What's impressive and concerning is how bacteria can develop these traits through mutations or by acquiring genes from others via horizontal gene transfer. Each of these strategies represents a different evolutionary solution to the same problem: survival in the presence of antibiotics. Let's start with perhaps the best-known resistance mechanism, enzymatic inactivation, particularly by beta-lactamases. Beta-lactam antibiotics, like penicillins, cephalosporins, and carbapenems, work by targeting bacterial cell wall synthesis. They do this by binding to enzymes called penicillin-binding proteins, which help construct the cell wall. Bacteria fight back by producing beta-lactamases, enzymes that hydrolyze the beta-lactam ring of these antibiotics, rendering them useless. There are several classes of beta-lactamases. Narrow spectrum, e.g., TM1, that break down penicillins. Extended spectrum beta-lactamases, ESBLs, that act on cephalosporins. Carbapenemases like KPC and NDM that destroy even our last-line carbapenems. These enzymes are often plasmid-encoded, making them easily transferable between bacteria. Clinically, this means infections that were once easily treated now require more toxic or less effective alternatives. For example, ESBL-producing E. coli and Klebsiella are a growing threat in hospitals. To counter this, we sometimes pair beta-lactams with beta-lactamase inhibitors like clavulanic acid. But bacteria are always evolving new beta-lactamases, staying one step ahead. Now, let's talk about another enzymatic resistance pathway, aminoglycoside-modifying enzymes, or AMEs. Aminoglycosides, like gentamicin, tobramycin, and amikacin, work by binding to the bacterial 30S ribosomal subunit and disrupting protein synthesis. They're potent, but bacteria have developed clever ways to neutralize them. There are three major types of AMEs. Acetyltransferases, AACs, add acetyl groups. Phosphotransferases, APHs, add phosphate groups. Nucleotidyltransferases, ANTs, add nucleotidyl groups. Each of these chemical modifications alters the aminoglycoside molecule so it can no longer bind effectively to the ribosome. These enzymes are often encoded on plasmids or transposons, making them highly mobile between bacterial species. You'll often find AMEs in gram-negative pathogens like Pseudomonas aeruginosa and Enterobacteriaceae. Some clinical isolates carry multiple AMEs, making them resistant to all aminoglycosides. This mechanism is especially problematic in ICU settings, where aminoglycosides are used as part of combination therapy for severe infections. So while aminoglycosides are powerful tools, bacteria have found biochemical ways to defuse them. Next up, target site modification, specifically involving penicillin-binding proteins or PBPs. PBPs are the molecular targets for beta-lactam antibiotics. They're involved in the final steps of bacterial cell wall synthesis. When a beta-lactam binds to a PBP, it prevents cell wall cross-linking, weakening the cell and leading to lysis. Some bacteria develop resistance by altering the structure of their PBPs so that beta-lactams can no longer bind effectively. A classic example is Streptococcus pneumoniae, which has modified PBPs that reduce penicillin binding affinity. Then there's MRSA, methicillin-resistant Staphylococcus aureus. MRSA acquires a new gene, MICA, which encodes a novel PBP called PBP2A. This protein has a low affinity for beta-lactams, rendering them largely ineffective. What's interesting is that these modified PBPs still carry out their function in cell wall synthesis. They just don't interact with the antibiotic. This mechanism makes infections like MRSA and penicillin-resistant pneumococci difficult to treat with conventional beta-lactams. Instead, we need to use alternatives like vancomycin or linezolid, which are more expensive and have more side effects. Understanding this mechanism helps explain why some beta-lactams work in certain cases but fail in others. let's explore another type of target modification, ribosomal protection. Many antibiotics target bacterial ribosomes to shut down protein synthesis. For instance, tetracyclines bind the 30S subunit, blocking the attachment of aminoacyl tRNA. This halts translation. Some bacteria have evolved ribosomal protection proteins, RPPs. These proteins physically dislodge the antibiotic or change the ribosome's conformation so the antibiotic can no longer bind effectively. One well-known example is TET-M, found in Streptococcus and Enterococcus. This protein allows the ribosome to continue translating proteins even in the presence of tetracycline. Ribosomal protection genes are often plasmid-encoded and associated with transposons, which means they can spread quickly between species. This mechanism doesn't destroy the antibiotic, it just renders it ineffective by shielding the ribosomal target. It's a clever adaptation. Rather than fighting the drug head-on, the bacteria make the target invisible or inaccessible. Clinically, ribosomal protection is an important reason why tetracycline resistance is so widespread, especially in community-acquired pathogens. It also highlights the evolutionary arms race between drug design and bacterial adaptation. Fluoroquinolones like ciprofloxacin and levofloxacin are broad-spectrum antibiotics that target DNA gyrase and topoisomerase 4, enzymes essential for bacterial DNA replication. Resistance arises primarily through point mutations in the genes encoding these enzymes, specifically GYRA and PARC. These mutations alter the drug-binding site, reducing the antibiotic's ability to inhibit enzyme function. The result? The antibiotic still enters the cell, but can no longer stop DNA replication. The bacteria continue to grow and divide. Additional resistance can come from increased expression of efflux pumps or decreased outer membrane permeability, further reducing intracellular drug concentration. This is particularly common in gram-negative pathogens like E. coli, Klebsiella, and Pseudomonas, but also in Staphylococcus aureus. Clinically, fluoroquinolone resistance is troubling because these drugs are often used empirically for UTIs, respiratory infections, and gastrointestinal diseases. Treatment failures can be serious and lead to complications. This mechanism also demonstrates how a single-point mutation in just one gene can render an entire drug class ineffective. Now, let's discuss efflux pumps, one of the most versatile resistance mechanisms. Efflux pumps are protein complexes embedded in the bacterial membrane that actively export antibiotics out of the cell. This reduces the intracellular concentration of the drug, allowing the bacteria to survive. There are 5 major families. Major Facilitator Superfamily, MFS. ADP Binding Cassette, ABC, Transporters. Resistance Nodulation Division, RA, Family. Small Multidrug Resistance, SMR, Family. Multidrug and Toxic Compound Extrusion, MATE, Family. One classic example is Acarabitol-C in E. coli, which belongs to the R&D family. It exports multiple classes of antibiotics, including beta-lactams, tetracyclines, and fluoroquinolones. Efflux pumps can be either specific or broad-spectrum, and their expression can be constitutive or induced by antibiotic exposure. Overexpression of efflux pumps is a major reason for multidrug resistance, MDR. When combined with other resistance mechanisms like enzyme production or target mutation, the result is a pathogen that's nearly untreatable. Efflux pumps also complicate drug development. Even if an antibiotic can bind its target, it may not be effective if it's pumped out too quickly. In summary, efflux pumps are the bacterial version of a force field, blocking drugs not by neutralizing them, but by expelling them before they can do harm. Now, let's explore another powerful resistance mechanism, reduced permeability, particularly through the loss or alteration of porins. Porins are proteins located in the outer membrane of gram-negative bacteria. They act like gatekeepers, allowing small hydrophilic molecules, including many antibiotics, to enter the bacterial cell. When bacteria lose these porins or mutate them, antibiotics can no longer effectively penetrate the cell. This drastically reduces drug accumulation at the site of action. One well-known example is in Klebsiella pneumoniae and Pseudomonas aeruginosa. These bacteria often develop resistance to beta-lactams and carbapenems not only by producing beta-lactamases, but also by downregulating or mutating porin proteins like OMK35 and OMK36. The combined effect is devastating: low permeability and high enzymatic destruction. Porin loss doesn't usually cause resistance on its own, but when combined with other mechanisms, especially efflux pumps or enzymatic degradation, it contributes to high-level multidrug resistance. Understanding porin-related resistance is essential because it also helps explain why some antibiotics fail even when no conventional resistance genes are detected. It also complicates drug development. How do we design molecules that can still enter cells with fewer doors? The takeaway: sometimes resistance doesn't come from what the bacteria do, but from what they stop doing. In this case, they simply stop letting the antibiotic in. Now, let's focus on how resistance spreads at the genetic level, specifically through mobile genetic elements. Resistance genes are often found on mobile DNA elements like plasmids, transposons, and integrons. These elements can move between bacteria, even across species, facilitating the rapid spread of resistance traits. Plasmids are small, circular DNA molecules that replicate independently of chromosomal DNA. They often carry multiple resistance genes. For example, a single plasmid can carry genes for beta-lactamase production, efflux pumps, and aminoglycoside-modifying enzymes. Transposons and integrons play supporting roles. Transposons can jump from one piece of DNA to another, spreading resistance genes within or between plasmids and chromosomes. Integrons collect and express gene cassettes, often including resistance determinants. Horizontal gene transfer via conjugation, plasmid sharing, transformation, uptake of free DNA, or transduction via bacteriophages is the engine driving this genetic mobility. A well-known example is the NDM1 gene, which encodes a carbapenemase. It's spread rapidly across continents, largely via plasmids, turning previously treatable infections into nightmares. These mobile elements transform bacterial populations. They don't need to wait for mutations, they can just borrow the tools for resistance. This is why infection control and surveillance are so critical. Once a resistance gene is out in the wild, it's hard to reel it back in. Let's zoom in on one of the most concerning pathogens today: carbapenem-resistant Klebsiella pneumoniae. Pneumoniae is a gram-negative bacterium commonly found in the human gut but capable of causing serious infections, particularly in healthcare settings. When it becomes resistant to carbapenems, our last resort antibiotics for gram-negatives, treatment becomes extremely difficult. The main mechanism here is the production of carbapenemases, enzymes that hydrolyze carbapenems. These include KPC, Klebsiella pneumoniae carbapenemase, NDM1, New Delhi metallobeta-lactamase, and OXA48. These genes are often carried on plasmids, making horizontal gene transfer easy and fast. Patients infected with CRE, carbapenem-resistant Enterobacteriaceae, can suffer from bloodstream infections, pneumonia, and UTIs. Mortality rates can exceed 40 to 50%, especially in immunocompromised individuals. Outbreaks in hospitals are hard to control. CRE can survive on surfaces and spread via healthcare workers' hands. Infection control measures, hand hygiene, screening, cohorting patients, are essential. Treatment is challenging. Older drugs like colistin or tigecycline are used, but they have high toxicity and limited efficacy. Newer combinations like ceftazidime-avibactam offer some hope but are expensive and not always accessible. Pneumoniae CRE is a clear example of how mobile genes, enzymatic resistance, and hospital ecology combine into a public health crisis. Why does resistance evolve in the first place? It's all about selective pressure and survival advantage. In the presence of antibiotics, susceptible bacteria die off, leaving behind those with resistance traits. These resistant bacteria then multiply and, over time, dominate the population. This is natural selection at work. Even small amounts of antibiotic, like those found in wastewater or livestock feed, create selective pressure. Bacteria exposed to sublethal doses may not die, but the stress can trigger mutations or activate mobile elements, increasing the likelihood of resistance development. Furthermore, bacteria replicate quickly and in huge numbers. That gives them a statistical edge. Mutations are rare, but when you have billions of cells, even rare events happen often. Add in horizontal gene transfer, and you've got a recipe for rapid evolution. A good example is fluoroquinolone resistance in E. coli. A single-point mutation in DNA gyrase can reduce drug binding. Add another mutation and an efflux pump gene, and resistance jumps significantly. The key concept: bacteria don't want to become resistant. But under pressure, the ones that randomly gain resistance survive and thrive. And with each treatment failure or misused dose, we increase that pressure. Let's now turn our attention to where resistance genes come from in the broader ecosystem. Antibiotic resistance doesn't just emerge in hospitals. The environment is a huge, often overlooked reservoir. Antibiotics and resistant bacteria enter the environment through various pathways: wastewater from pharmaceutical factories, agricultural runoff, and even the feces of humans and animals. These compounds and organisms mix in water bodies and soil, creating a microbial melting pot. Bacteria in these environments, many of them non-pathogenic, exchange resistance genes through horizontal gene transfer. These genes can then be picked up by pathogenic bacteria. For instance, resistance genes found in clinical strains of E. coli and Salmonella have been traced back to soil bacteria. A key example is tetracycline resistance in agricultural settings. Tetracyclines are used in livestock, and resistant bacteria in manure can transfer their resistance to other bacteria in the soil and water. Eventually, these bacteria can colonize humans through food or contact. What's worrying is that environmental bacteria often harbor novel resistance genes that haven't yet emerged clinically. So, the environment acts as both a reservoir and an incubator. Understanding this environmental dimension is crucial for any long-term strategy to tackle resistance. It also calls for a One Health approach, integrating human, animal, and environmental health policy. We need to think of resistance not just as a medical issue, but as an ecological one. Now, let's talk about what happens when bacteria accumulate multiple resistance mechanisms, so-called superbugs. Multidrug-resistant (MDR) organisms are resistant to 3 or more antimicrobial classes. Extensively drug-resistant (XDR) and pandrug-resistant (PDR) strains take it even further, leaving us with virtually no treatment options. Common MDR superbugs include Acinetobacter baumannii, often resistant to carbapenems, aminoglycosides, and fluoroquinolones. Pseudomonas aeruginosa, capable of porin loss, efflux pump overexpression, and enzyme production. Klebsiella pneumoniae, frequently harbors extended-spectrum beta-lactamases, ESBLs, and carbapenems. Staphylococcus aureus. MRSA strains often also resist macrolides and fluoroquinolones. These pathogens thrive in hospital environments. ICU patients, especially those with invasive devices or prolonged antibiotic exposure, are most at risk. Superbugs represent the convergence of multiple resistance pathways: enzymatic destruction, target modification, efflux, and decreased uptake. That's why tackling them requires combination therapy, strict infection control, and sometimes last resort drugs with serious side effects. The rise of superbugs is a warning. It tells us that resistance isn't theoretical, it's already undermining modern medicine. Now that we understand the problem, what can we do about it? First, antibiotic stewardship is crucial. This means prescribing antibiotics only when truly necessary, selecting the right drug, dose, and duration. Stewardship programs bring together microbiologists, pharmacists, and clinicians to oversee and optimize antibiotic use. Second, infection prevention, hand hygiene, sterilization, and patient isolation can reduce transmission. It's simple, but incredibly effective. Third, innovation in drug development is critical. We need new antibiotics that work through novel mechanisms. We also need to think beyond antibiotics. Bacteriophages, antimicrobial peptides, and CRISPR-based tools show promise. Fourth, Public education and global cooperation are essential. Resistance spreads across borders and sectors, so solutions must be interdisciplinary and international. And finally, we need better diagnostics and surveillance to track resistance and guide therapy in real time. Together, these strategies form a firewall against resistance. But they require constant maintenance and global commitment. What if we could not just stop resistance but reverse it? That's the idea behind resistance reversal. One approach is beta-lactamase inhibitors like clavulanic acid or avibactam. These are given with beta-lactam antibiotics to block the enzymes that would otherwise destroy them. Another strategy is efflux pump inhibitors. These compounds block the bacterial pumps that expel antibiotics. While none are in routine use yet, research is ongoing. CRISPR-Cas systems offer a high-tech option. They can be programmed to target and cut resistance genes in bacterial genomes or plasmids, effectively de-resisting the population. Collateral sensitivity is a fascinating concept where resistance to one drug makes bacteria more vulnerable to another. If we can map these relationships, we might design treatment cycles that select against resistance. Finally, microbiome restoration using probiotics or fecal transplants may outcompete resistant pathogens in the gut, especially after antibiotic therapy. These approaches are still in development, but they highlight an important shift from playing defense to going on the offensive against resistance. Let's wrap up with a few key takeaways. First, antibiotic resistance is a multifaceted problem involving molecular biology, ecology, medicine, and public policy. There is no silver bullet. Second, bacteria use a variety of mechanisms to resist antibiotics: enzymatic inactivation, target modification, efflux, and permeability changes, and they can gain these traits rapidly via mutation or horizontal gene transfer. Third, the clinical and environmental consequences are severe. From treatment failures to environmental reservoirs, resistance is reshaping modern medicine. Fourth, we must act on multiple fronts: stewardship, innovation, policy, and global cooperation. Novel therapies and diagnostics offer promise, but without behavior change and regulation, they won't be enough. And finally, as microbiologists and healthcare professionals, you are at the forefront of this battle. Your knowledge, vigilance, and innovation are crucial. Thank you for your attention, and more importantly, for your commitment to tackling this critical challenge.